The cause of individual differences in behaviour and personality is one of the most hotly debated subjects in the history of psychology. Over time, theories have shifted between those which emphasise the role of the environment in moulding the character of a person, and those which propose that differences between people are largely innate. At one extreme lies behaviourism, which posits that people are like blank slates upon which their character is written by the environment, and at the other, genetic determinism, the idea that individual differences in psychological characteristics are determined completely by genetics.
Francis Galton, great-grandson of Erasmus Darwin and second cousin of Charles Darwin, was the first to have attempted to study the inheritance of behaviour empirically. Inspired by the magnitude of his cousin's scientific breakthrough, he became obsessed with studying, in his own not-so-humble words, the heredity of greatness. Among his observations, he noticed that some sets of twins were identical, whereas others were only as similar as regular full siblings. He took several measures of physical and psychological characteristics of identical and non-identical twins, and from his results, he estimated that identical twins were around twice as similar as non-identical twins. Since the environment was assumed to be equally as similar for identical and non-identical twins, Galton derived that around half of the variance in behaviour can be accounted for by heredity.
Although his experiments had some holes in their methodology, Galton's estimates turned out to be impressively accurate for someone who had no knowledge of DNA. Unfortunately, for all of Galton's genius, inherited or otherwise, he had one very bad idea which tainted his legacy: eugenics.
Galton believed that civilization could be bettered by improving the human stock, by giving the more suitable races or strains of blood a better chance of prevailing speedily over the less suitable than they otherwise would have had. This idea spread and gained popularity among political parties across Europe and North America. Eugenics programs provided positive enforcement, such as financial incentives to those deemed most fit for reproduction, and negative enforcement, including forced sterilisation of those deemed unfit, such as those with disabilities, low IQ scores, criminal records, and minority racial identities. This was carried out in countries including the USA and, most notoriously, Germany under Nazi rule.
As a result of the horrors of the 20th century, the public and academic institutions alike, quite understandably, developed a distaste for studies into the heritability of psychological traits, and the subject was shelved.
This created an academic vacuum ready to be filled with environmentalist theories of human nature, such as Sigmund Freud's theory of psychosexual development and B.F. Skinner's radical behaviourism. Psychology remained in the grips of environmentalism until the 1960s, when this consensus was challenged by a new subfield of genetics called behavioural genetics and a novel methodology which was beginning to stack up mountains of evidence to the contrary.
Behavioural genetics is a field of study which aims to discover the influence of genetics on behaviour.
In this regard, it is similar to the related field of evolutionary psychology. However, where evolutionary psychology investigates the reasons why differing behaviours evolve in different species, behavioural genetics aims to quantify the contribution of genetics to individual differences in behaviour within the same species. The genetic influence on individual differences is estimated by a statistic called heritability. Heritability describes the proportion of the variance of a trait within a population which can be attributed to genetics. This is not the same as saying that heritability measures how much of the trait is caused by genetics. Rather, it describes how much of the differences in a trait between individuals in a population is caused by genetics. Heritability estimates are only just that, estimates, the accuracy of which can be affected by factors such as similarity of the environment and age of testing of the trait. Heritability estimates paint a picture of the genetic contribution to individual differences in traits of the population they were tested in. However, heritability estimates of the same trait may vary between populations in different environments. An example of this is the fact that heritability of body weight is higher in more affluent countries than in poorer countries. This is because, in poorer countries, people are more restricted in their food consumption because of lack of availability, so their body weight will be more dependent upon how much nutrition they have access to than their genetics. Whereas in affluent countries, where almost everyone can afford to eat more than they need, body weight will depend more on genetically influenced factors such as metabolism, frame, and appetite.
Another caveat is that heritability describes the correlation of genetics with traits, not causality. Heritability tells us nothing about the pathways from genetics to behaviour, and it often takes tortuously indirect routes.
For example, attractive people tend to score higher on measures of self-confidence than average, presumably as a result of being more well received socially. Part of the heritability of self-confidence can therefore be explained by genetic variants which, rather than affecting brain areas associated with self-confidence as one might assume, result in prominent cheekbones and acute button nose. Heritability is estimated by dividing the correlation of a trait in related individuals by their coefficient of relatedness. This is easier to express as a formula, where T equals correlation of the trait and G equals the coefficient of relatedness. Heritability equals T over G. Correlation of the trait refers to how similar the trait is in genetic relatives compared to the population as a whole. For dichotomous traits, characteristics which a person either has or does not have, this is calculated simply by finding out how likely someone is to have a trait, such as a diagnosis of schizophrenia or a penchant for skydiving, given that a genetic relative, such as a sibling, cousin or parent, also expresses the trait. For quantitative traits, characteristics which every individual has but at varying levels, correlation of the trait is calculated by how accurately you can predict the level of a trait, such as height or IQ scores, in an individual, given that you know the level of the trait in a genetic relative, compared to how much the trait varies from the mean average in the population as a whole. The strength of the correlation is denoted by a statistic called R², the value of which can vary between 0 and 1. A correlation of R² equals 0 means that the pairs of genetic relatives are no more similar to each other than any pair of individuals in the population as a whole, whereas a correlation of 1 means that the trait of one of the pair of genetic relatives perfectly predicts the trait in the other. It is extremely unlikely that R² would ever be exactly 0 or 1 for any trait, rather it usually falls somewhere in between. The coefficient of relatedness is a measure of how closely related two people are and is roughly equivalent to the proportion of genetic variants which they had inherited from the same ancestor. So identical twins will have a coefficient of relatedness of 1, full siblings and parent-child pairs will have 0.5, grandparent and grandchild pairs, cousins and nephews will have 0.25, and so on.
This formula for estimating heritability works because the correlation of the trait describes how similar the genetically related pairs are, and, all else being equal, this similarity must be caused by their shared DNA. So, we can divide the strength of this correlation by the coefficient of relatedness of the genetically related pairs to discover what proportion of the variance of the trait is attributable to genetics.
To give a hypothetical example, if full siblings had a correlation of R² equals 0.25 in their extraversion scores, you could estimate that extraversion was 50% heritable by following the formula for estimating heritability, as 0.25 divided by 0.5 equals 0.5 or 50%. You could reach the same conclusion if the extraversion scores for full cousins had a correlation of R² equals 0.125, or if the correlation was R² equals 0.5 for identical twins.
Obviously, there are confounding factors which need to be considered when designing experiments for determining the heritability of traits. The most relevant being that related people tend to share similar upbringings as they usually grow up in the same family households, so the similarity in traits could equally have been caused by similar upbringing. This problem is tackled in part by the use of adoption studies.
Adoption studies work by comparing the similarity in traits between adoptees and their biological family members with that of their adoptive family. For example, measuring extraversion in adoptees and comparing the similarity between their extraversion scores and that of their birth parents with the similarity between their extraversion scores and that of their adoptive parents. A similar method is comparing the similarity of adoptive siblings to that of biological siblings. Adoption studies mimic the well-established animal research technique of cross-fostering, where animal offspring are removed from their biological parents at birth and raised by surrogates to investigate genetic-linked behaviours and physical traits. Another method of addressing the shared environment issue is by studying twins. Identical twins have been called God's gift to genetics because they share 100% of their DNA, allowing the opportunity for a natural experimental control. Twin studies work by comparing the similarity of pairs of monozygotic, also known as identical, twins with that of dizygotic, also known as non-identical or fraternal twins. Because both kinds of twins share very similar environment, including gestating in the same womb and being born on the same day, any difference between the similarity of monozygotic twins and that of dizygotic twins must be in the most part caused by genetics.
Neither of these methods are infallible on their own. For example, in twin studies, there is the assumption that identical twins are treated equally as similarly as fraternal twins, despite some evidence to suggest that this is not the case. And in adoption studies, middle-class families are overrepresented relative to the population as a whole, potentially resulting in an overestimation of heritability since environmental variance is restricted. But, by compiling the evidence gathered from several methods, researchers can account for the weaknesses of each with the strengths of others and triangulate upon more and more accurate estimates of heritability.
A situation which offers the best of both from the two aforementioned methods is cases of identical twins separated at birth. Heritability can be estimated by comparing the variance of a trait between identical twins separated at birth with that of identical twins raised together. As monozygotic twins are genetically identical, any difference in similarity between the twins raised apart and the twins raised together must be caused by the environment. Cases of identical twins separated at birth are unfortunate for the people involved and are thankfully vanishingly rare. They are also, however, very useful when it comes to the study of nature and nurture, so behavioural geneticists try to document as many of such cases as they can find. You might have already heard about twin studies or anecdotes about the remarkable similarity of twins. Twins separated at birth are often discussed in the media, highlighting astounding similarities in behavioural quirks between separated twins. The Jim twins, so called because their adoptive parents both named them Jim, who were separated at birth and reunited in 1979 at the age of 39, are among the most famous examples. As well as being remarkably similar in their appearance, they also both enjoyed mechanical drawing as a hobby, preferred the same subjects at school, had both married women named Linda, divorced them, remarried a woman named Betty, and both had sons whom they named James Allen.
Anecdotes like these are remarkable, however, they do not tell us much about the genetics of human psychology. Not much that is quantifiable anyway. The real discoveries from twin studies are to be found in traits measured in objective and standardised pen and paper tests carried out on large sample sizes.
One such category of psychological traits which can be objectively measured is personality. Personality traits are habitual patterns of behaviour, thought and emotion that are stable properties of an individual's psychology. There are 5 dimensions of personality agreed upon by most personality researchers. These include openness, conscientiousness, extroversion, agreeableness and neuroticism. Intelligence is another measurable aspect of psychology. As well as quantitative dimensions of psychology, taking yes-no measures of life events which are in large part a result of the choices made by the individual, such as obtaining a university degree, being divorced, having a criminal record or seeking support for mental health, form another route to investigating individual differences in psychology.
The psychologist Thomas Bouchard, who had heard about the Jim twins and invited them to be the subject of his research, started receiving correspondences from other such separated twins soon after publishing the results of his case study. He eventually collected data from over 100 pairs of twins. Having a sample of this size created the perfect opportunity for the genetics of individual differences in psychology to be studied. By providing a reference point for genetic similarity and removing shared environment from the equation, for the first time in the history of science, twin and adoption studies had made it possible to disentangle and empirically measure the respective contributions of nature and nurture, and nature was coming up larger than anyone had expected.
The findings of twin and adoption studies have been summarised in the 3 laws of behavioural genetics. All behavioural traits are heritable, the effect of genetics is always greater than the effect of shared environment, there is a substantial proportion of the variance in behaviour not explained by shared genes or families.
The most startling discovery brought about by twin and adoption studies, described in the first law, is that there is not a single known measurable behavioural characteristic which does not show any genetic influence. Every psychological characteristic that has been investigated in twin and adoption studies has been found to be correlated between family members, and the strength of this correlation increases with the percentage of shared DNA. Not only are all behavioural traits heritable, the second law tells us that genetic factors always have a greater influence on behaviour than the home environment. Behavioural traits in adoptees are always on average more similar to their biological family than their adopted ones. Therefore, the effect of genetics on behaviour must be greater than the shared environment. The third law describes the fact that there are no behavioural traits which are accounted for completely by the combined influence of heritability and the home environment. Identical twins who grow up in the same home are very similar but not psychologically identical. Therefore, there must be other factors affecting behaviour independent of genetics and the shared environment. Behavioural geneticists refer to this as the non-shared environment, the things which we experience in life which are unique to us as individuals. There are therefore three categories of factors which influence our behaviour: heritability, the shared environment, and the non-shared environment. The influence of the non-shared environment can therefore be calculated by subtracting heritability, as estimated using the methods described previously, from the correlation of a trait in identical twins sharing the same home environment. It turns out that the effect of the shared environment is close to negligible. This is evidenced by the fact that in measures of behaviour, siblings raised apart are no less similar than siblings raised together, and by the fact that adopted siblings are no more similar than strangers. The most generous estimates on the impact of the shared environment is around 10%, however most estimates are closer to zero. The bulk of the non-genetic influence on behaviour is therefore made up by the non-shared environment. The non-shared environment is comprised of the non-systematic events which happen to us in our life which are unique to us as individuals. Catching a virus as a newborn, a neighbour who teaches you how to play chess, a sporting injury, a musical instrument discovered in the loft, and everything in between. Their effect on our behaviour is like that of a pinball game, nudging us in different directions as we move through life. The non-shared environment is mysterious, hard to identify and measure, non-stable and random. It is for these reasons that, despite explaining a substantial proportion of the variance in behaviour between individuals, the non-shared environment is not a useful predictor of behaviour. It is just too unique to each individual to be quantified. Findings of behavioural genetics have been solid, despite the harsh scrutiny the studies had been placed under due to the controversy surrounding the topic. In fact, the statistical rigour with which behavioural genetic studies have had to be carried out under to stand up to such scrutiny has resulted in behavioural genetics being one of the modern psychological theories which has survived the so-called replication crisis, in which more than half of the findings of papers published in highly regarded academic journals turned out to be statistical flukes. We must bear in mind, however, the caveats about heritability mentioned earlier. Heritability estimates are only applicable to the population in which they were calculated. So, although being raised in a different home environment within the same culture is not likely to make much of a difference to your behaviour, that is not to say that being raised in a radically different culture would not have had an impact. It is entirely possible that a person would have ended up with a different personality had they grown up in the Amazon jungle rather than the City of London. Another point to bear in mind is that estimates of the shared environment are made under the assumption that there is at least adequate nurture in the home environment. Cases of abuse, neglect and deprivation do have an impact on normal psychological development. Despite these reservations, the findings from twin and adoption studies are transformative for the way we think about ourselves. The finding that DNA is a much more important and stable influencing factor on who we are than the way we were raised developed a fatal blow to the doctrine of radical behaviourism, which had psychology in its grips for the latter half of the 20th century. People have their own genetic selves, which, given adequate nurture, they may flourish into. Just as a gardener may create the right environment for flowers to bloom, but cannot change the type of flower, a seed will become.